I'm going to talk today about BioStudies, which is a new database of biological studies, and I work at the EBI, which is the European Bioinformatics Institute in Cambridge in the UK. So, as you may know, the EBI offers a number of free global databases, including Ensemble, ENA, Pride, and Array Express, which is to name just a few. But the concept of BioStudies is to be a single collection point for all the data about a study, regardless of where that data for the study resides. And BioStudies will simplify linking to data from research articles, as well as improve the discovery of related data across data resources. So that's the concept of BioStudies, but how are we actually sort of aiming to achieve this, and what we actually want to achieve. So we want to be able to link multi-omics, we will be able to link multi-omics studies. Currently, if scientists carry out a project, they might do some sequencing work, and they'd have to deposit their data at ENA, they might do some protein work, and then that data would have to go to Pride, and there's no sort of central linking of the metadata to describe that studies. And that's what we'll be able to offer. And it's not about just linking with internal resources, but it's also about being able to link to external resources. So we currently have had a study submitted to us which links to the image data repository. The obvious advantage of this is that the EBI holds many petabytes of information, and it's hardworthy and expensive centrally storing this data. And as we can store this loose and almost unstructured data, it means that we're not constrained by the formal archives. This is particularly advantageous as technologies improve and the data standards can't quite keep up. So we'll be able to accept these sort of new data, this new data. And we've already been able to do this with a person who was a submitter that couldn't, their data wasn't able to fit within the ENA, and we could have provided them with an accession number, which meant they were then able to submit to a journal. And of this flexibility with being able to archive this data means that we can work with our collaborators such as DICSA, which represents toxicological studies, and we can represent this data sort of very specific to that project, such as compound views. So, although we're a new database, it doesn't mean we're empty. We've currently got 500,000 studies within. That's because we've been working with the Euro PMC, our colleagues there, which is a literature archive, which is the European version of PubMed. And what we want to do is provide a one-stop shop for this supplementary material and manage the supplementary files and links that accompany publications which are currently unarchived. So I told you about the data that we hope to contain, and obviously people will need to be able to access this data. So we offer a number of sort of computational access, such as XML and APIs to access it computationally. You'll also be able to download the data with FTP. And we also have been developing our user interface on the web, and within that we've built on the sort of previous searching tools that are used by traditional databases, and we've also been able to represent files in a number of different views and be able to sort within those files, and if we have a table that's a file, it's being able to display and you can sort within that table. And I've also talked about, there's not just about viewing, it's also about submitting. We're going to currently launch a tool. This will be self-curating, which means that you won't need to involve a curator. You'll be able to amend. And finally, I'd like to say, if you would like any more information, please come and phone me on post row 12, and I'll be at the EBI stand booth 10, and I'll acknowledge my team up there. Thank you.
Very much.